despite my excitement, and I think it would be fair to say the field's excitement about the new era of precision medicine in the face of genomic sequencing, there are some real challenges that us as clinicians are going to have to grapple with in the next decade. And I think that's the increasing realization that tumours are phenomenally complex. And I say that because of the complexities we're seeing in the genomic data that's coming through. And to cut a long story short, what we're seeing is that tumours are essentially evolving like trees, and that sounds like a bit of an odd analogy, but we have the trunks of the tumour's tree, which is the early events in tumour evolution that trigger cancer cell growth. And those events may be mutations in particular genes that drive cancer growth from a very early stage. And those early mutations are carried through from daughter cell to daughter cell to daughter cell. And propagated throughout the entire cancer mass, and those mutations are called, trunk mutations. But later on in tumour evolution, as the tumour grows larger and cells separate, new mutations can occur in some cells but not others, and we call those subclones. And I picture those as sort of the branches of the tumour's evolutionary tree, where individual branches depict individual cancer cells or in individual regions of the tumour that may be separated both spatially within a tumour, but also temporally and change over time. And so we have the sort of aspects of tumour evolution to deal with where a static snapshot point in time genome sequence of a tumour may not tell you terribly much about what's going to happen to the tumour in a year or two's time. And that's why we're going to have think about ways of having continuous monitoring of the disease. And research is going to be so vitally important here, because I think we're increasingly seeing that tumour evolution is not random. Is actually quite constrained. And my sort of nirvana, if you like, is hoping that we'll see a future where we'll be able to predict the next evolutionary move a tumour is going to take and stop it from happening in advance of it happening, rather than doing what we're doing in the clinical situation at the moment, which is reacting to tumour change after it's happened.